Okay, so we'll begin immunity today. So let's look at the different types of immunity that our body provides us with. The first one is what is known as non-specific immunity, or also known as innate immunity. And here we have two lines of defense, because as any pathogen wants to enter our body, so you have these forces. So first naturally would be anything which, the first thing that the pathogen or whatever substance is trying to get inside your body is going to come in contact with. So it's going to be your external covering, like the skin, or your internal lining, like a mucous membrane. Okay? For example, if something was to poke your eye, or something was to go into your nasal cavity, or something, you were to swallow something, so then that would be the lining of the gastrointestinal tract, or the nasal mucous membrane, or the mucous membrane of your eye, like the conjunctiva. So that's called the first line of defense. So it's a physical barrier that is present. So skin and mucous membranes come under this. And the way they help is they produce a variety of substances. So let's look at the skin first. So the first thing is, it is the skin, the epithelium itself is very thick. So it is stratified squamous, keratinized epithelium. So remember, the keratin present here is an important barrier to preventing anything from entering. So it's a thick epithelium. Then the skin also produces sweat, which has a low pH. So it's acidic in nature. It has some bactericidal properties, so it prevents growth of microorganisms. So, when you sweat profusely, it kind of washes off these microorganisms which may colonize on the surface of your skin. So that helps. Sebum, which is produced by the sebaceous glands, that also helps. Then let's look at mucous membranes. So in your mucous membrane, that is the lining epithelium, let's see some of the things which are mentioned here. So one is acid, for example, acid in the stomach, which is, again, maintains the low pH, so it prevents colonization of bacteria. Then you have substances called lysozymes, which also have what are known as bactericidal properties, so they kill bacteria. These are produced in your saliva, tears, mucus. Then you have goblet cells and simple columnar cells which produce a lot of mucus. That mucus in your mucous membrane, that is very thick. It is able to trap substances which may go there. You also have the production of molecules called defensins, which again help to prevent bacterial growth. So these are some of the things that skin and mucous membranes do. If you also think of it, your urine, just by when you cry, tears, not only do they produce lysozymes, but tears also wash away cell debris. So does urine, for example. It washes away anything that might be. So that's why, they tell you not to hold your urine for too long, because the purpose is to flush away anything that might, if urine is stagnant, the bacteria can colonize there. So that we don't want that to happen. So urine again helps to flush out, bacteria or anything that might be present there. So this is the first line of defense. The second line of defense is comprised of either cells or some chemical substances which are secreted. So let's look at the cells which take part in the second line of defense. The first are cells which are capable of phagocytosis. In order to be able to phagocytose something, you need to have a lot of cytoplasm. The cells that are capable of phagocytosis, you've done some of these in blood. So neutrophil is one. Monocytes are another. Monocytes change into macrophages. So, and then we have other macrophages which are present. You have others in the brain, for example, you have microglia. In the skin, you actually have cells called dendritic cells, or also known as Langerhans cells. Dendritic cells or Langerhans cells which are present in skin. So all of these are phagocytic, and what they do is these phagocytic cells, so imagine this is a cell and this is a bacterium or something which is trying to invade, the cell throws out these little processes which are known as pseudopodia, or like feet. It engulfs the bacterium which goes inside, and then inside the cell you have certain suicide sacs which are called lysosomes. So these are different from lysozyme. Lysosomes are suicide cell organelles which have digestive enzymes, and then they're capable of digesting this. So this is what phagocytosis does. Then another kind of cell we have which are lymphocytes. These are known as natural killer cells. So what they do is they don't phagocytose, but inside them, these lymphocytes, they have a number of granules. And these granules secrete something known as perforins and other substances. So what they do is that they attach themselves to a cell which has been infected by a virus. So imagine that this is a cell which has a virus inside, or maybe it's a cancer cell. So what they do is when they attach themselves to these virus-infected cells or cancer cells, they secrete these perforins, and as the name suggests, perforin means to perforate. So they cause minute holes in the cell membrane. So the cell actually just ruptures or lyses. So there is cell lysis. So that's the way the natural killer cells act. Then in the body there's a process called inflammation, which we'll do in the next slide. So I'm going to skip to the next one and then come to inflammation later. So then we said second line of defense was cellular or chemical. So there are certain antimicrobial proteins. So these are chemicals which are secreted. One of them is known as interferon. So it's a protein which is secreted by cells which have been infected by viruses. Now those cells cannot save themselves, but what they do is that they secrete this interferon, and what interferon does is viruses survive in us by replicating their DNA, and then one virus becomes millions of viruses, and then they can go and invade other cells. So what interferon does is that while it is produced by a virus infected cell which cannot save itself, it now goes out and it prevents the virus from replicating. So the viruses then cannot enter other cells and they cannot undergo replication, which is their way of spread of the infection. So they're not able to do that. And the other thing that interferon does is it also activates natural killer cells, which will come on the scene and then they can cause lysis. And it also causes macrophages to come to that area, activates macrophages. So again, by phagocytosis, they can take care of those cells. So this is how antimicrobial protein interferon acts. Then we have another kind of protein. There are actually 20 of them. These are known as complement proteins. And these complement proteins, they are in an inactive form in our body. They need to be activated. And they take part not only in this non-specific immunity which we are talking about, but they also take part in specific immunity, which we'll see later. So when antibodies and antigens combine, they can activate this complement protein. So it's present in an inactive form. It needs to be activated. And when it is activated, what it does is it causes the cell to lyse. The cell ruptures. So imagine this is a cell which needs to be taken care of. So these complement proteins, they actually kind of make a little tunnel like this. And they burrow into the cell, and the cell ruptures this way. It completely sort of, bursts. So that is what is called cell lysis. And the other thing that complement does is it also promotes phagocytosis. So it brings about these other cells which can do phagocytosis, like neutrophils, macrophages. It brings them to that site and helps with phagocytosis. So that's how complement proteins act. And then another line of defense is that your body produces, when it is infected, it produces substances which are known as pyrogens. These raise your body temperature. They act on the hypothalamus. and they raise your body temperature. That's why high fever is known as pyrexia, from the word pyrogen. And what fever does is it makes substances unavailable, substances like iron and zinc. It makes it unavailable to the bacteria and other pathogens which need them to survive. So it makes this unavailable. Because your body temperature is high, it increases metabolism and that promotes repair. So fever actually is a good way. It's a way of the body trying to heal itself. The problem comes when your body temperature goes beyond a good value. You know, it becomes very, very high, 103, 104. When it's really high, then it interferes with enzyme systems, because remember, high temperature denatures enzymes. So then is when you want to bring it down. But if you have low grade fever like 100 or a little bit more than that, you don't always have to take some medicine to bring down the fever, because it's really kind of helping you. So here now let's look at the way complement acts. So I told you, remember, it created a tunnel. So let's look at the way complement will act.
The membrane attack complex is part of the complement system, which exists to aid in inflammation, phagocytosis, and other aspects of the immune defense, including pathogens from the body. There are two primary stages in which the complex is formed. The first is...
Can you hear that? Were you able to hear that? Not really. Okay, let me try this.
The membrane attack complex is part of the complement system, which exists to aid in inflammation, phagocytosis, and other aspects of the immune defense, including pathogens from the body. There are two primary stages in which the complex is formed. The first is referred to as initiation. During initiation, protein C5b binds C6 and C7. The newly formed C5b67 complex binds to the brain by. The second stage, through which the complex to pathogen, is referred to as the polymerization stage. C8 binds to the complex and is able to insert itself into the membrane because of traits relating to the polarity of the molecules that make up the membrane and the protein itself. C8 is then able to induce many C9 proteins to form into a porous structure that penetrates the cell surface, filling it with water and oils, inevitably destroying the cell.
So you don't have to worry, they keep mentioning C7 and C6 and all that, but you saw how, they kind of one sat on top of the other and they ultimately call the entire cell to rupture. So they kind of make a burrow into it and make a hole in it. So let's look at some questions here. So which of the following are produced by the skin to provide the first line of defense? Okay, it is keratin and sweat. So it's B and C. See, the skin does not produce mucus. Think of it. Does your skin produce anything slimy? Mucus, it doesn't. See, mucus is produced by mucous membranes, by those goblet cells or columnar cells. Your epithelium here, it does not produce mucus. Sweat glands produce a very thin watery fluid, and sebaceous glands produce sebum, which is oily. They are oil glands. Okay, so mucus is not produced by the skin. It's produced by the mucous membranes. So it was B and C. So this one. So here let's look at inflammation. So what happens in inflammation? So imagine something has disrupted the surface of the skin. And so here there are inflammatory chemicals which are, sort of poured out from that area. And neutrophils come, the phagocytic cells, of which neutrophils are one. They come, the moment they sense that some damage has been done, they come to that area. The capillaries which are flowing through the dermis of the skin in that area, since we're talking about skin at this time, the capillary permeability increases. So fluid begins to sort of seep out. That causes a bit of swelling as fluid seeps out. these, you can see these neutrophils which are present here. These neutrophils, slowly what they do is they, from the center, they come to the side of the blood vessel and they leave the blood vessel by a process which is known as diapedesis, and they go towards the area where those inflammatory chemicals are being released, and that is called chemotaxis. Now, because of the swelling in this area, because fluid has seeped out, that area gets swollen. You've all noticed, if you had something bit you, or you can see that that area gets raised. It's red in color, feels a little warm. It's swollen. You might feel pain, right? So, because the capillary permeability increases, fluid seeps out. That causes swelling. Because of the swelling, the nerves could get compressed, and that causes pain. Because the blood vessels dilate in that area, that causes the redness that you see in that area. And whenever blood vessels dilate, more blood is flowing through that area, so that will raise the temperature. So these are 4 cardinal signs of inflammation, which in Latin were known as rubor, which is redness, and that is due to vasodilation. Calor means temperature. So rubor was redness. Temperature is raised because there's more blood flowing through that area. Tumor means swelling. Swelling is because the capillary permeability has increased, so fluid has gone out. And dolor is pain. The pain is because those nerve endings have been compressed. So now when all this happens, what happens in that area is that wherever the inflammation is going on, and you know for yourself, you've all had at some point even a little mosquito bite, you get a little sort of a wheal in that area swelling, or you've injured yourself somewhere and you find a little inflammation. So can you see the inflammation is localized? So that part is swollen, it's just localized. So the idea is to localize it, remove the injurious stimuli, and then start the healing process. Okay, so that's the purpose of inflammation. So let's look at how this happens.
Inflammation is the protective reaction of vascularized tissue to local injury. Acute inflammation is the early, almost immediate reaction of tissue to the injury, and it is the first phase of wound healing. Acute inflammation can be triggered by cell or tissue damage, or by the presence of dead cells or noxious agents such as bacteria. Acute inflammation typically occurs before the immune response becomes established and aims primarily at removing the injury-causing agent and limiting the extent of tissue damage. Acute inflammation occurs in 2 overlapping stages, vascular and cellular. In the vascular stage, arterioles and venules near the site of injury constrict briefly, then dilate. Dilation is congenital, while an accompanying increase in capillary permeability leads to movement of fluid into the affected tissue, resulting in the signs of inflammation: redness, swelling, heat, pain, and loss of function. As fluid leaves the capillaries, the blood remaining in circulation becomes more viscous. It flows more slowly, and clotting occurs. The cellular stage of acute inflammation is initiated by the movement of phagocytic white blood cells, or leukocytes, into the area of injury. The leukocytes begin to adhere to the vessel wall, and then, in a process called emigration, squeeze through the wall and move into the inflamed tissue. The leukocytes wander through the tissue, guided by chemical signals in a process called chemotaxis. The cellular stage culminates in the leukocytes engulfing and degrading the bacteria and cellular debris in a process called phagocytosis. Products of phagocytosis, along with plasma and blood cells, form exudates, which accumulate, causing swelling, and exudates are composed of serous fluid, red blood cells, fibrinogen, or tissue debris, and white blood cell breakdown products. Concurrent with the events of the vascular and cellular stages, chemical mediators, bioactives that act to mediate the inflammatory response, derived from the cells or from plasma. One of the first mediators of an inflammatory response is a cell-derived mediator, histamine, found in high concentrations in the mast cells of connective tissues adjacent to blood vessels, as well as in blood basophils and platelets. Histamine is released in response to a variety of stimuli and causes dilation and increased permeability of capillaries. Serotonin, another cell-derived mediator, performs similar actions. The three major plasma-derived mediators are present in the plasma in precursor forms that must be activated, usually by a series of proteolytic enzymes. The kinins increase capillary permeability and stimulate pain receptors. The clotting system traps exudates, microorganisms, and foreign bodies. The complement cascade causes vasodilation, promotes leukocyte chemotaxis, and augments phagocytosis. Acute inflammation comprises a variety of complex protective responses to injury that together lay the groundwork for the next stages of the body's recovery: immune response and tissue repair.
So you don't need to know the measures and so on. You can see how those were released, and you saw how those neutrophils were kind of going through the process. Emigration is the same as diaphylesis, okay? They were attracted there. So let's answer some questions. So is this true or false? Yes, chemotaxis is attraction due to chemical. Taxis means movements, chemo is chemicals. So this was that's how the neutrophils went through that. So again, very important to look at the PowerPoint slides and look at the label. So you can see positive chemotaxis means, can you see the inflammatory chemicals here? And you can see these moving towards that. Okay, let's do a little review. Which of this is not seen in inflammation? Yes, release of mucus and lysozymes. You don't see that. We never mentioned anywhere in our PowerPoint slide or even in that video about mucus being released or lysozymes, which are, bactericidal enzymes which are released. We didn't talk about that, but everything else is present. Okay, which of these best describes the purpose of inflammation? Okay, very good. Yes. See, when you look at this, you must see what it says, which of these statements best describes. So see which statement kind of uses everything. So you do localize infection, and the idea is to promote repair or healing of the tissue. So yes, cell debris is removed, infection is localized, but to localize and remove all this, the idea is to promote repair. So this is the one which best describes what's going to happen. So here is a really good slide taken from your book. So I'm not going to go over that. So you can kind of go through this and see, what the first line of defense, like, so for example, it tells you about the skin and, the mucus, skin and mucous membrane. So, when you look at this first line of defense, remember skin and mucous membrane. So, these two things, see what all the skin does, how the skin secretions help, keratin, what it does. And then the mucous membranes are the ones who produce mucus. Then what I didn't mention was even mucous membranes in places they have cilia, and in your nose, for example, they have nasal hairs. Those also help by their movement, so they trap cell debris or push them. For example, in the respiratory system, the cilia in the nasal cavity and pharynx beat down, whereas from the trachea they beat upwards, so that they don't allow it to go into the lungs. In acid, we talked of in the stomach, but you also have it in many areas where the skin also has a low pH, the vagina also low pH, so it prevents bacterial or microorganisms from colonizing that area. And then we talked about all this. And even urine is acidic normally, and it cleanses as it flushes out the system. This is the second line of defense, and again you can see these are cells or chemicals, so you can see phagocytes, natural killer cells, inflammatory response, then these are the proteins. So this is kind of like a quick look over everything, so be sure to look at this slide when you're reviewing for your quiz and test. So based on this, a foreign body enters your eye and irritates your conjunctiva, causing you to tear up. So what line of defense has been breached? So I just took you through that review. So think of it, which line of defense has been breached? First line of defense, think of it, conjunctiva is mucous membrane, okay? So when your eye, the lining over here, like you don't have skin lining your eye, but you have, this conjunctiva, which is the mucous membrane. So when something goes into your eye, the first thing it comes in contact with is this protective layer, the conjunctiva, okay? So second line of defense, remember, were chemicals and they were cells. So this is mucous membrane. So first line of defense. Again, some more to review. Which of the following statements is incorrect? Yes, remember cells was second line of defense. So here it says phagocytic cells are neutrophils and macrophages. They are second line, not the first line of defense. Everything else is the first line of defense. So what we did first was what was called innate or non-specific immunity. So now we are going to look at what has often been also called the third line of defense and known as specific immunity. And what you must realize right from the outset is that it isn't as if the body can only use one. So it always starts with the first line of defense tries to prevent something, then the second line of defense helps it, and then it might go on to the specific immunity, or it may go directly to specific immunity. It could be anything. Sometimes you'll see, as we'll see in later slides, all of them act together. So everything helps to make sure that the immune system is functioning in the best possible manner. So in specific immunity, the first thing is that it is of two types. One is called humoral, where you have the production of antibodies. And the second is called cell-mediated, where it is a cell-to-cell reaction. So it's like one cell latches onto the other and destroys it, much like the natural killer cells. Only we don't use the natural killer cells in specific immunity, and you'll see later why. So here what happens is the cells, and it's the lymphocytes which take part in this specific immunity. You have B and T lymphocytes. The B lymphocytes take part in humoral immunity and the T lymphocytes in cell-mediated, and there are different types of T lymphocytes. So in order for this to happen, the lymphocytes have actually got to be introduced to the antigen. That is what is called priming. So it's like, imagine you go to a party, you don't know anybody, and there's an introduction made, there's a little bit of time it takes for you to warm up to them. So same way with this, since the antigen first has to be introduced, then the cell has to react. So it takes time to mount the immune response. So this specific immunity is also called adaptive immunity. This takes longer to mount an immune response. Plus, we call it specific, so it must be something specific to it. It is antigen specific. So in other words, there are lymphocytes in your body, for example, which would react, let's say, to the chickenpox antigen. So if the chickenpox virus was to come inside your body, these lymphocytes would be able to react. But another set of lymphocytes, which say were specific to the measles antigen, they cannot react to the chickenpox virus. So can you see it's very antigen specific. So that's again a very, very important thing in the specific immunity. So it's specific. It doesn't mean it can react to anything that comes in, but only to a specific antigen for which it was in fetal life, it had been programmed to react against. This is not local. Remember, we saw non-specific, like for inflammation, for example, it tends to be localized to an area. This, in specific immunity, it's systemic. So it kind of spreads throughout the body. And one good thing about it is that this has memory. So once it's been introduced, it's like if you went to a party and you were introduced to someone, and you kind of remember that, and the next time you see that person, it triggers a memory, and you kind of react a little quicker. You don't take that long to open up a conversation with them. Same thing with specific immunity, has memory. So the first time it reacts to the antigen it was programmed to react with, it takes a while. But what it does, when it's doing its job, it kind of remembers it, and you produce cells which are known as memory cells. So the second time the same antigen comes into the body, the reaction is much faster because now it knows this antigen. So this memory can last for a very, very long time. And that's what happens when you take vaccinations, or you don't fall ill for a really long time, or you say you contracted chickenpox when you were a child, and then you don't know, you've probably been exposed to the virus all through, but it's because of those memory cells which are there in your body which recognize that antigen, and they react to it without you ever manifesting the disease. So this memory cells helps us to respond and bring about a quick, rapid response to the antigen that it is supposed to react with. So now let's define antigens. So antigens are substances which are usually protein in nature. Most of them are protein in nature, but they may be large carbohydrates or polysaccharides, some lipids, and sometimes nucleic acids. And these are capable of provoking an immune response. So we did blood, and if you remember, you looked at antigen A, which decides that the person's blood group will be A. Antigen B, blood group B, or a positive Rh, the D antigen, which makes the person Rh positive. Remember, if they were introduced into a person into the wrong blood type, remember how they provoked a reaction? So that's what is the purpose of an antigen, to provoke an immune response. The antigens are seen on the surface of cells, on the external surface of cells, and the red blood cells was a very good example. Remember, the antigen A sitting on the top of the cell gave the blood group A. The antibodies, if you remember, were present in the plasma. And then inside us, we have certain protein markers known as MHC, or Major Histocompatibility Complex. MHC stands for Major Histocompatibility Complex. These are protein markers on the surface of your cell. So here is this protein marker up here. So imagine this is the MHC protein, this one. There are two types, 1 and 2, but you don't need to go into that. These display on the surface and they tell the body that it is self. Or MHC proteins from another organism would be non-self as far as we are concerned. And then they also display antigen. So they display self or non-self antigen. So here is the MHC marker, which you can see. And then it displays the self antigen. So inside every cell of your body, there are many antigens present. Those are processed and broken down into smaller molecules. And then this MHC acts like a marker and, flags it on the surface of the cell. Imagine another small little foreign antigen comes into your body, it gets into the cell, then inside the cell that is kind of broken down and processed, and now the MHC marker displays this non-self also. So in your body, your lymphocytes during fetal life are programmed in such a way that when they see a self antigen and a self MHC protein, they kind of ignore it because they say, oh, this one is from part of the family, we won't do anything to it. But when they see a non-self, because they have been trained to react only to non-self, which is what you want them to do. So when they see that non-self antigen being displayed over there, so then what they will do is they will react to it and they will destroy, they'll produce the immune response. Now there are certain antigens in your body which never come in contact with blood. So with the result that if ever they did come in contact, they can provoke a response, an immune response. Even though they belong to self, but because they never come in contact with blood, they are kind of kept in isolation. So these are known as sequestered antigens. And examples of sequestered antigens are the lens of your eye and even the sperm, the human sperm. This becomes important. You've all heard when people have cataracts and you remove the lens, you all normally want the cataract to ripen. You want it to become hard. You don't want to remove it, when it's soft, because if it ruptures, part of it could get in contact with the tissue of your eye and it could set up a violent reaction because, see, the body recognizes it as foreign then. The same with sperm. When you do vasectomy, you always want to make sure that the ductus deferens, which carries the sperms, so this is the duct, you want to kind of tie it off at the two ends and make sure that these two ends are tight, no sperms leak out, because if the sperms leak out, again they could set up a violent reaction. Okay, so these are two examples of what are called sequestered antigens. So let's look at the cells now of the immune system and also see how the immune system develops. So the first are the cells, and APC stands for antigen presenting cells. These are antigen presenting cells. Examples of antigen presenting cells are dendritic cells in your skin, those Langerhans cells that I talked of, macrophages, and some B lymphocytes. So what these antigen presenting cells do is actually, they will take an antigen, they will get an antigen, process the antigen, and they will show it on the surface of the cell, of their self. And then these are then recognized. You'll see these antigen presenting cells when we talk about T lymphocytes. Because T lymphocytes cannot recognize antigens which are free. They have to be present on the surface of a cell. So these antigens get into the body. The APC cell picks them up, processes it, raises a flag to show that this is a foreign antigen, and then those T lymphocytes react against them. Okay? The other immune cells are the B lymphocytes, which take part in the humoral response. And then we have T lymphocytes, which take part in cell-mediated response. And there are three types of T lymphocytes. One is called cytotoxic, as the name suggests, toxic to cyto or cell. These are the ones which carry out the actual destruction of the cell. So the actual cell-mediated response is carried out by these cytotoxic cells. These are also known as effector cells. Then we have something called helper T lymphocytes. You will see later these helper T lymphocytes are the most important cell in the immune system really, because they stimulate the cytotoxic as well as the B lymphocytes. They are what trigger these two to clone and make more cells. And then we have another group called suppressor, or also often known as regulatory T lymphocytes, which are lymphocytes which will turn off the immune system, because you don't want the immunity to go on and on and on. The response to keep continuing. If it goes on and on and on, you would actually end up disrupting homeostasis. So once the effect that you want has been achieved, you need these cells to turn off the immune system. So they are known as regulatory or suppressor T cells. So let's look at how the immune system develops in the fetus. So in the fetus, in the bone marrow, where all the cells are being formed, the blood cells, so WBCs, RBCs, and platelets. So there now, part of the WBCs, the lymphocyte precursors are developing and they are forming in the bone marrow. Those lymphocytes which have to become T lymphocytes, what they will do, they will migrate to the thymus. That's why they are called T lymphocytes, or thymus-dependent lymphocyte. And those lymphocytes which have to become B lymphocytes will remain in the bone marrow, and then they will mature in the bone marrow. They are called B lymphocytes, so they mature in the bone marrow, and that's how you can remember it, though it's not from, they're not called B lymphocytes because they're destined for the bone marrow. It comes from something known as bursa of Fabricius. So this bursa, that's why they were called B lymphocytes. So lymphocytes which have to become T lymphocytes, these precursors will go to the thymus, and those which are destined to form B lymphocytes will remain in the bone marrow. Now when they go into the thymus, they will start maturing and they achieve what is known as immunocompetence. That means they get training. They are trained to become competent to take part in the immune response. So it's like going to military school for training, okay? So that, imagine you go to West Point to get trained to be a soldier. So that's what they're doing. And the B lymphocytes remain in the bone marrow and they become immunocompetent. And they also develop something known as self-tolerance, which I'll just show you in the next slide. Because you don't want, these lymphocytes are naive. So these are naive. That means to begin with, they have not yet become trained. So they can attack any and everybody. They can just run loose. So you want cells, these lymphocytes, to develop into lymphocytes which will recognize self and not attack them. You want them to be able to recognize non-self. And then, in other words, they have to be tolerant to self and intolerant to something which is foreign. Isn't it? That's what you want to kind of achieve in the end. So when they go in over here into the thymus and the bone marrow, they achieve immunocompetence, and they also achieve self-tolerance. So if all those lymphocytes which would react against self, what happens is they die. So a lot of cell death occurs, because these cells, if they are allowed to live, what will happen? They'll begin to attack your own cell. So they are killed off. All those cells which can react against foreign antigens, means antigens which don't belong to you, they are kept, because that's what you want, right? You don't, if you go, imagine a military school, anybody who would be a traitor to the country, you don't want to train them. Those who will fight for your country, you want to train them. So that's exactly what's going to happen. And sometimes there are some lymphocytes which are weakly reactive. That means they are iffy. They may or may not. So some of them are kept in the body and they are made self-tolerant. So they are sort of trained that, don't react against self and you need to be tolerant towards self. After they mature in the thymus and the bone marrow, then they go out into the general circulation, where they begin to go into all the lymphatic structures which you just saw. You remember all the areas, the lymph nodes, the tonsils, the Peyer's patches, all of those areas. So they go there. And then, let's say at one point in time, let's say we are talking about a B lymphocyte over here. So a B lymphocyte now is still what is known as naive. Imagine a soldier who's been trained in a military base or something. He still hasn't had the experience of battle. So he's still naive. He's competent, but you won't know how competent he is till he actually goes into battle, right? So these lymphocytes are immunocompetent, but they're still naive. That means they still haven't had any encounter with an antigen. So now these, when they encounter an antigen, so here say a B lymphocyte, it has these receptors. Remember, they were antigen specific. So these receptors will only react to the antigen that it has been programmed for. Remember, I told you that they're all programmed for different antigens. And you know, you can think about, imagine the antigens to be like the alphabet. You can make different combinations. So genes determine how many antigens our cells, our lymphocytes are programmed to react against. So we have so many antigens all over the world, floating around, and our body cells are capable of reacting towards most of them. It just so happens that not necessarily that you would come in contact with all of their antigens throughout life. So let's say I have a cell in my body which is capable of reacting against yellow fever. But if I never come in contact with the yellow fever virus, that lymphocyte would forever, it's going to die with me. So it would never have come in contact with that. So can you understand that? So similarly, in your body, there are your self-antigens which are part of that. So those lymphocytes have to be, those which react against those self-antigens, those have to be killed before they are thrown out into the circulation. So they have to make sure that they only react against those group of antigens which don't belong to self. Okay? So here a lymphocyte has this receptor. It comes in contact with the antigen that it was programmed, it was trained to react with. So then it becomes activated. And what it does is, this is a single lymphocyte. So one lymphocyte is not going to be able to do the job against that antigen, right? So then what it does is, the moment it comes in contact with the antigen, it undergoes cloning, or what is called proliferation. So this is the general process that happens in the immune response. It undergoes cloning or proliferation. So the activated lymphocytes, they multiply into numerous cells. So you have more of these cells which are exactly like this. That means they can now recognize that same antigen and act against it. So of these clones which are formed, some of these clones will form memory cells. Some of these clones will form memory cells, and most of them, so let's say a whole bunch of them, will form what are known as effector cells. So this happens both for B and T lymphocytes. And the effector cells are the ones which will actually carry out the immune response. The memory cells will stay in your blood and keep circulating, so that the next time that same antigen comes into your body, they'll recognize it faster, multiply again, form more memory cells, and then the effector cells will take care of the immune whatever response has to be done. Now this is how it happens in your body. How is this selection process occurring? How does the body decide which lymphocytes to keep during, when they are being trained in the bone marrow and thymus, and which lymphocytes to kill off? So the first thing is what is called positive selection. So they use T cells because it has been studied more in T cells than in B lymphocytes. So first of all, the T cells, they must be able to recognize the self MHC proteins. So here, if this is a T cell receptor, if it cannot recognize a self MHC protein, it will die, because it's like you can't recognize the self protein. There's no point in you living, so they kill it off. Those lymphocytes which can recognize the self MHC protein, remember those markers, they are allowed to live. So that's the first step. So this is called positive selection. Now, remember those MHC proteins carried these antigens on them. So this was the protein part, this purplish color, and this orange is the antigen. So first thing they have to do is they have to recognize this purplish part, the protein. Now, the next thing is that remember I told you that these MHC proteins carry self-antigens on them or non-self antigens on them. So, if these lymphocytes, they recognize the self-antigen, that means they are going to react against it. So they are killed off. So you understand now it is negative selection. So they must not recognize self-antigens. In other words, they must be tolerant to self-antigens, okay? So those cells which recognize self-antigens, they are going to react against them, so they will be killed off. Those that don't recognize self-antigens, that means they are tolerant to them, they will then be allowed to live. So that means they are able to recognize foreign antigens, and they can, take care of the situation. Okay, so that's what happens in selection. Now, sometimes you may have heard of a situation known as autoimmunity. Now, in autoimmunity, what happens is that something goes wrong in the immune system. So here, in autoimmunity, this recognition apparatus breaks down. So with the result, the lymphocytes begin to attack their own body cells. It could be due to a number of reasons. One could be maybe some of these cells were not weeded out. These cells which are reactive against self-antigens, maybe they were not weeded out. Maybe some of those iffy cells, remember I mentioned there were some which were iffy, which were supposed to become self-tolerant. Maybe they didn't become self-tolerant. Or you could have, nowadays there's so many pollutants in the environment which cause mutations. So maybe a self-antigen mutated in such a manner, or sometimes there are certain substances which resemble these antigens and they kind of combine with them. So the body thinks of it as foreign and begins to attack it. So a variety of reasons why autoimmunity could occur. And here the body begins to attack its own tissues. So there are a number of autoimmune diseases that are present. So can you think of any? Any autoimmune disease? None? No, there's a difference between AIDS and autoimmunity. So AIDS is not the same as autoimmunity. In autoimmunity, the body attacks its own cells. In AIDS, what happens is the immune system is deficient. That's why it's called acquired immune deficient syndrome. The immune system is deficient because the virus is killing off some of the helper T cells. So it's not that the body is attacking its own cell. Your immune system is so deficient that it cannot do its job properly, so you're falling prey to a lot of other infections, okay? So examples of autoimmunity are rheumatoid arthritis, type 1 diabetes, myasthenia gravis. Remember I told you about this in the thymus. Psoriasis. So many there. I mean, every day they are kind of finding so many of these diseases which, are autoimmune. There's a disease called sarcoidosis. A lot of the connective tissue disorders are autoimmune. Lupus, you may have all heard of lupus. That's again autoimmune, okay? So the body is not able to recognize its cells and instead it begins to attack them. So let's identify the incorrect statement here. Okay. Okay, very good. Yes, those that react against self-antigens are not kept under surveillance throughout. Like they are destroyed right away. Imagine, it takes too much trouble for the body to keep these guys under surveillance. Imagine if you had people in a country who were going to be terrorists to your country. You're not going to kind of keep them under surveillance, right? You're going to, well, probably not kill them, but you want to keep them away, right? Okay, let's see what are MHC proteins. So I shouldn't have had the R over there. MHC proteins. Yes, very good. They process and display antigens on cell surface. Okay, what are sequestered antigens? Yes, they are self-antigens that have never come in contact with blood. Not that they are antigens which were introduced into the body at one given location. These were, remember, lens and the sperm. They are part of your body, but they just didn't come in contact with blood. So let's look at the first of the specific responses, which is the humoral response. So some things to remember in the humoral response. The first is it's carried out by B lymphocytes. And these B lymphocytes, you'll see, when they clone and they multiply, they form effector cells, which are called plasma cells. So these are the effector cells. And then they form memory B cells, which will react faster during a secondary response. What B lymphocytes do is they produce antibodies. So this is humoral response is when antibodies are being produced. And these antibodies, they bind to extracellular targets. So an antigen like here, for example, you have this free-floating antigen which is here. Okay, it could be anything, a bacterium, a virus or something. So it's lying free. The antibody will come and attach to this antigen and it will form an antibody-antigen complex. And then, you know, it will take care of the problem. So you can see that it is outside the cell. In cell-mediated, what will happen is if this is a normal body cell, the virus will go in and it will go inside the cell. And now a cytotoxic T lymphocyte, after going through a certain variety of processes, it will react against this. So can you see it's a cell-to-cell reaction? Okay. So let's see what happens in humoral immunity. So first is what is called a primary response, which always takes long. Remember when we talked of specific immunity, we said it takes long to mount a response because you have to be first introduced to it. So the primary response is the first encounter with the antigen. The secondary response is any subsequent encounter after the primary. So here's an antigen, and here are many lymphocytes which are floating around. And these are the different receptors which are present on these lymphocytes. Okay, so they've just shown you different colors. There's a green, a blue, and a dark blue and a light blue. Okay, so let's say this one reacts to chickenpox virus, this one reacts to the measles virus, and this one reacts to yellow fever. So let's say the measles virus gets into the body, and so that's it on itself. The virus itself is an antigen. So now it can only attach itself to that cell which was programmed to react with it and has the receptor for measles virus. So you can see that it binds here. These two can do nothing, so they just ignore it because they don't have the receptor for it. So it binds to the specific B lymphocyte which is capable, which was coded to react with it. The moment it binds to it, that B lymphocyte becomes activated. It immediately starts multiplying. And so it forms many cells which are exactly similar to it. So they all will have receptors which can react with those kind of, with that same measles virus. So when it multiplies and forms similar cells, we call it cloning. So they form a clone. So the clone that is formed, lots of cells are formed. So in this, so it will first form, say, 2, 4, and they go on forming, and you form a whole bunch of cells. Of these, most of them will become what are called effector cells, or they are also known as plasma cells. And a few of them will be known as memory B cells. So you don't want all of them to become memory B cells, because, memory is for reaction at a later time. Effector is for reaction now. So here you have many of these plasma cells and a few memory cells. So these are primed to react to the same antigen. These plasma cells, they secrete these antibody molecules. So you can look at these antibody molecules. These antibody molecules have, as you can see, it looks like they have the same structure as these receptors. The function of these molecules is they are capable of reacting to the antigen the same way that receptor was capable of doing. So what these antibody molecules will do is they will react with that antigen, because there are many. It's not like one bacteria only gets into your body or one virus, right? So many get in, and when they react with this, they form antigen-antibody complexes. Let's see what happens to this memory cell. So this plasma cell, it secretes these antibody molecules, they react, and then the plasma cells go off. Now let's say many years later, this memory cell it still lives. It travels in your body for a long time. It lives for a really, really long time. This, say again you were exposed to the measles virus. And now that memory cell, it quickly recognizes that antigen, and the response is much faster, and it produces again cloning. So of this, many of them become plasma cells. Again, you have some few which will become memory cells. So can you see that the memory cell line keeps on being propagated, so that you don't have only one set of memory cells who have to do the job all the time. So all through your life, you're constantly being exposed to these infections. You never realize you were exposed to it, because the plasma cells have taken care of the infection. The time when you do get measles is when these plasma cells, they did produce antibodies, but it was not enough to completely sort of squish that infection, so you manifest with the disease. Okay? So sometimes you may have had the infection, you just don't realize because you didn't come up with the disease. So these antibody molecules are secreted, and they react with the antigen, and they'll form antigen-antibody complexes, and memory cells will continue to be produced. In the second response, if you have another exposure, again memory cells will be produced. Now there are different classes of antibodies, and antibodies, another name for antibodies is gamma globulins. Or also called immunoglobulins. You might hear of it as immune serum sometimes. Immune serum means serum which contains this gamma globulins or immunoglobulins. So we have different classes of antibodies, and they are produced at different times. So the first one is Ig, stands for immunoglobulin A. This is produced on the body surfaces, in breast milk, and so on, IgA. Then we have something called IgD. They are present on the surface. Previously, the function was not known. Now they think they are present on the surface of cells, acting as antigen receptors. So they're present on the surface, acting as receptors. IgA was, as I said, present in breast milk, surface of skin. So they act as antigen receptors. IgM is produced in the primary response. IgG is the secondary and subsequent response, usually secondary and subsequent, and this is the most plentiful. And IgE is usually seen in allergies, hypersensitivity reactions, some types of hypersensitivity reactions. So these are the different classes of antibodies. So let's see what happens when an antigen and an antibody get together. They form what is called an antigen-antibody complex. These are removed from the body. The antigen-antibody complex are protein molecules. It's not a cell, right? It's a protein molecule. So you've got to get rid of it. So it's removed from the body, and there are different ways that it's removed, and we often call it a plan of action. So let's see how this happens. So here you can see, here's the antigen, here's the antibody. Together they meet and they form a complex. This plan of action is, the P stands for precipitation. So they kind of get precipitated and then they're removed by, either they can be removed by phagocytosis, usually by phagocytosis, or what can happen is that the antigen-antibody complex can activate the complement protein, and that complement protein, if you remember, it causes cell lysis, or it also helps in phagocytosis. So that's the L, lysis. So P stands for precipitation. P is the precipitation. L is lysis. So you can do it this way, P, L, A, and N. So I won't write it. So that's the plan. A stands for agglutination. So what they can do is they can cause the cells to get stuck to each other, which you saw whenever they're in mismatched transfusion. You saw how red blood cells agglutinate with, if they have the antigen and the antibody. Or they can, antigen-antibody complexes also undergo neutralization, where they, some dangerous parts of the bacterial exotoxins or viruses are masked so that they don't produce their deleterious effects, their harmful effects. So this, if you look at this picture, you can see an antigen-antibody complex. What happens? It gets inactivated either by precipitation, agglutination, or neutralization. These in turn now have to be removed, so phagocytosis is the best way to remove them. Or it can activate complement. Remember, I told you complement took part in specific and non-specific immunity. We already saw it in non-specific. Here you can see in specific how it gets activated by antigen-antibody complexes. The complement leads to the cell being destroyed, plus the complement also causes phagocytosis and inflammation. So you can see that this is how antigen-antibody complexes, antigen-antibody complex acts. Okay? Sometimes the antigen-antibody complexes may be so large that when they get precipitated, for example, if you have a very bad, massive transfusion reaction, I told you when we were doing blood, the hemoglobin gets, because of lysis of the cell, that hemoglobin can go and deposit in the kidney, and it can prevent the filtration, or it can clog the filtration membrane, and the person can undergo renal failure. So sometimes when you have a very, very massive reaction, some problems can arise because of that. So let's answer this question. Which of the below statements is correct? all of the above are correct. So humoral immunity does involve cloning of B lymphocytes. It does involve the production of antibodies. Plasma cells take part because they are the ones which produce the antibodies. It is antigen specific. Remember, all specific immunity is antigen specific. So all of this is important. This one. True, yes.